Welcome to today's lesson on cloning methods, where we will uncover the processes and implications of replicating organisms. In today's lesson, we will explore various methods of cloning in both plants and animals. Additionally, we will discuss the potential benefits and risks of cloning in agriculture and medicine, and consider the ethical objections some people have towards these practices. So far, you have learned about genetic engineering, which has provided you with a foundation in understanding how we can manipulate genetic material to produce desired traits in organisms. This knowledge of genetic engineering sets the stage for today's discussion on cloning, as cloning leverages similar principles to create genetically identical organisms. Understanding the methods and implications of cloning is a crucial step in advancing our biological understanding, because it offers insights into how we can replicate organisms for agricultural improvements, medical treatments, and conservation efforts. However, it also raises important ethical considerations that we must contemplate. Let's get started. Plant cloning can be done from cells or sections of a parent plant. There are multiple ways to do so, including tissue culture and using cuttings. Tissue culture is a sophisticated method of plant cloning, allowing the reproduction of plants from just a few cells of a parent plant. Here's how it works. A few cells are taken from the parent plant. These cells are placed in a sterile petri dish containing a nutrient-rich growth medium. Specific hormones are added to induce cell division and growth. Over time, the cells grow into a mass of tissue. This tissue then develops into small plantlets. The plantlets are transferred to potting trays, where they mature into fully grown plants that are clones of the parent plant. Tissue culture is highly efficient, requiring minimal space and allowing for year-round cultivation. It's particularly useful for preserving rare plants and rapidly producing large quantities for nurseries. It can prevent the spread of diseases by producing native plants in high quantities without importing them. Cuttings are a more straightforward and traditional method of plant cloning. Healthy sections of a plant are cut. These cuttings are then planted in soil or water. The cuttings develop roots and grow into new plants. This method is quick and cost-effective and is commonly used by gardeners. Animal cloning is a more complex process, as differentiated adult animal cells do not readily divide. Instead, cloning techniques involve using embryo transplants, or insertion of an adult cell nucleus into an egg. Embryo transplants are widely used to clone animals, especially in agriculture. Here is an example of how an embryo transplant would be used to clone offspring of cows with desirable traits. Sperm from a prized bull is used to fertilize an egg from a prized cow. The embryo is allowed to develop to a certain stage. Before the cells specialize, the embryo is split into several individual embryos. These embryos are then implanted into separate host cows. The host cows give birth to calves that are genetically identical clones. This method allows farmers to produce a large number of identical offspring with desirable characteristics annually. Another remarkable method of cloning animals is through adult cell cloning, famously used to clone Dolly the sheep. Here's how it works. The nucleus is removed from an unfertilized egg cell. The nucleus from an adult body cell, for example, a skin cell, is removed and inserted into the egg cell. An electric shock stimulates the egg to start dividing and form an embryo. The embryo is then implanted into a surrogate mother's womb. The surrogate mother gives birth to an animal that is a clone of the donor adult. Now let's look at the benefits and risks of cloning. The benefits are that it produces large numbers of ideal offspring quickly. It also helps in preserving endangered species by increasing their numbers. Finally, it enhances our understanding of embryo development, aging, and age-related disorders. However, there are a number of risks. These include reduced genetic diversity, which can make populations with clones vulnerable to diseases. Furthermore, cloned animals may suffer from various health problems, such as Dolly the sheep, who developed arthritis. There are also significant ethical issues around human cloning and the possibility of numerous failed attempts before achieving success. Before we conclude today's lesson, let's review some historically significant cloned organisms.
One of the most pivotal cloning milestones was, of course, the birth of Dolly the sheep in 1996, whom I worked with. Dolly was the first mammal cloned from an adult somatic cell through a technique known as somatic cell nuclear transfer. However, she wasn't the first cloned animal. Scientists had successfully cloned frogs decades earlier, with the first amphibian cloning occurring in the 1950s. But Dolly's success was groundbreaking because it demonstrated that this technique could be extended to mammals. Her birth ignited a wave of ethical and scientific debates, reshaping the conversation about the possibilities and implications of cloning.
Here is a timeline summarizing the cloning advancements just discussed. In today's lesson, we explored cloning methods in plants and animals. We began with plant cloning techniques, including tissue culture, where small cell groups grow into new plants, and cuttings, where parts of a plant are replanted to grow new plants. We then moved to advanced methods like embryo transplants, where a fertilized egg's embryo is divided and implanted into host mothers, producing genetically identical offspring. We also covered adult cell cloning, highlighted by Dolly the sheep, where an adult cell's nucleus is inserted into an enucleated egg cell. We discussed the potential benefits of cloning, such as producing desirable traits and aiding medical research, along with risks like reduced genetic diversity, ethical concerns, and possible long-term impacts. This knowledge provides a solid foundation to understand the complexities, applications, and ethical debates surrounding cloning in science and society.